Okay, hello everyone, and it's a great honor to be here, and I want to thank the organizers for inviting me. And what I'll tell you about today is that there are head direction cells, not just in rodents, also in other animals, in bats. They're not rodents, although they're called flying mice in some languages. Not true. And that they're tuned to three-dimensional head directions, so direction there or there, as I'll show you. So 3D compass, if you will, and I'll also tell you about a different kind of directional representation that we found in the hippocampus that relates to vectors towards goal, direction and distances towards goal, which I would argue is crucial if you want to navigate and get somewhere. So we've heard a lot today about these various kinds of cells: place cells, grid cells, head direction cells, and border boundary cells. And these were typically, are often recorded in either kind of empty boxes in randomly foraging animals or on short tracks. And kind of provocatively, you might ask, how does real-life navigation differ from navigating in a 1 by 1 meter empty box? So we can come up with all sorts of answers, but these are my three answers. One is the world is not flat, it's complex and three-dimensional. And the world is also not empty, it contains goals. We navigate somewhere. So how does that represent in the brain? And the world is not 1 by 1 meter in size. Navigation occurs over kilometers. And so I wrote these particular three answers because we're actually directly addressing these two issues experimentally. So this is the animal we're working in. I'll actually leave to the end to justify why bats. I think it's easier first to show what we're doing and then to explain why this animal. And just by looking at its eyes, you'll believe me that they see very well. They're not blind at all. A quarter of mammalian species are bats. None of them is blind. These guys see really well. They have acuity half of that of a cat, or so much better than a rat. And they're also big, so we can, compare to bats at least, 150 to 180 grams, the big males, so we can put a lot of equipment on them. Neural loggers, GPS loggers, all sorts of equipment, and recording flying animals. And just to put you into the world of bats, what does it mean to navigate for an animal like that? We've done some years ago a study where we put GPS loggers, little devices just glued on the back of an animal in the wild. So these are wild bats. We're just behaviorally tracking their navigation behavior outdoors. And this is a typical track by one animal where you can see it leaves the cave here. The blue triangle goes to a local tree for a few minutes and then flies extremely straight to these two trees, feeds there on fruit for the entire night. These are fruit bats, so they're feeding on fruits there. And then it flies extremely straight back to the cave. So these guys can really navigate long distances. You can see the scale bar there. This is another example just to show they're not doing it for only one night, they're doing it on multiple nights. This is a navigation behavior for one animal over 7 nights in a row, so different colors are different nights. You see the animal flies from the cave to the same individual tree night after night. So these guys can really navigate, and we've done some experiments where we've translocated them and had them fly back, and that we argued that they have something like a map-like representation of the environment over large scales of roughly 100 kilometers. So this is eventually what I'd like to understand. How does that work in the brain? We're not there quite yet, but working on it. So this was my plan for half an hour. Then I decided it's a bit too ambitious. So we talk about 3D spatial codes, all of these types of cells, and goal-direct navigation, and about where we're going with a neural base of large-scale navigation. So yesterday I decided to just focus on something that I can cover reasonably in limited time. So I'll talk about 3D head direction cells and about goal-directed navigation using pictorial representations. So these are the students involved in the study of 3D, Michael Yartsev, Gilly Ginosar, and Arseny Finkelstein. So to start off with, over several years, we've recorded in bats crawling in two-dimensional boxes. We kind of ratified the bats in crawling in a two-dimensional box with a white cue card. Very much the kind of approach pioneered by Jim and his students. And we found similar kinds of cells as in rodents, so place cells, grid cells, maybe border cells. We haven't done yet kind of wall insertion experiments, but we have things that look like that along several walls or one wall, and head direction cells in this flat environment. A lot of the properties were similar to rodents. One major difference was that we did not see continuous state oscillations in in the bat. Actually, it's the only slide I'm going to show about that. For lack of time, I won't go into the detail, but you see on top are examples of two autocorrelations of rat neurons that I just took from the Moser website, beautifully oscillatory. At the bottom are interneurons from the hippocampus of the bat, and you see they're very flat. These are called theta cells, to remind you, in rodents, and there we were able to show that, looking at 2D place cells, 3D place cells, grid cells, interneurons in flight and in crawling, 160 cells, 0 out of 160, were significantly oscillatory at any frequency. So very clearly non-oscillatory. However, I'll just kind of tell you in half a minute without showing you the data that we do see some other interesting things, that although the LFP is extremely non-periodic, we see that the neurons do phase lock to this non-periodic LFP. So there's a synchronization of neurons. And very recently, it's all unpublished, so I'm kind of just telling you what we see. Very recently, we found that we can see phase precession relative to this non-periodic LFP. So you can define cycle by cycle the phase, and you can ask, do spikes occur early and early as the animal moves from the place field? And the answer, at least in some of the neurons, is yes. So I think this is kind of an interesting story that, in the rodents, you have three things that are always co-occurring together: the almost perfectly sine wave oscillation, synchronization, and phase coding. And in the bat, we don't see the first one. We don't see a perfect sine wave, but you see the other two ones, the synchronization and the coding. So I think this is an interesting example of how comparative approach can tell us something about what are the key features of the system. So let me go back to 3D. You can ask, when you see a place field that looks like that in 2 dimensions, what is the 3D volumetric representation? Could be any of those or anything else. So to address this, we, together with Neuralinks, we developed some years ago this system that allows us to record spikes in flight. So these are the 4 channels of one tetrode recorded in flight. You can see you can record nice spikes. Worked by Michael Yartsev when he was in my lab. And we've put this artificial tree in the middle of a big flight room that we have. It's almost 5 by 6 meters. And then we track the 3-dimensional position of the animal in the room with 2 video trackers. And then we see cells like that. This is an example neuron where you see that it fires in a limited region of 3-dimensional space. So whenever the animal flies through a volume that's roughly spherical and roughly, in this case, 1 meter in diameter, the neuron fires. And we found that for 91% of the cells, this was not significantly different from a sphere, meaning that there's the same resolution in the X, Y, and Z dimensions. So 3D place fields are spherical in shape or isotropic. What happens with the head erection signals? So Arseny Finkelstein in the lab addressed this question. We use this 3D tracking head stage. It's basically the same idea as with the colorful LEDs, but you just do it various ways. The way we did, we almost tetrahedral arrangement of non-coplanar LEDs, and then you can track all the 3 Euler angles, the azimuth or yaw, the pitch, and the roll of the animal as it's behaving, and record in the dorsal percebiculum of the bat, so the same area where head erection cells were originally discovered. And what Arseny found was the following. So the 3 columns here are 3 different cells. The rows are 2 different sessions separated by an hour. And what you see is that the neuron on the left, so the color is the firing rate, and this is pitch by azimuth. And you see that the neuron on the left doesn't care so much about pitch, but it's tuned very nicely to azimuth, so fires in a certain direction. Let's call it north, 0. So this is similar to a classical head erection cell in the rat. But then this neuron doesn't care so much about azimuth, but it is tuned to pitch, so negative pitch in this case. And there are also conjunctive neurons that are tuned to both azimuth and pitch, like that one. And from time to time, you could see, not very often, neurons tuned triply conjunctive to pitch, azimuth, and roll, like this one. We found that about 35% of the azimuth-tuned cells, the horizontal ones, about 35% were also tuned to pitch or roll, primarily to pitch. So the roll tuning was generally much weaker. So these are neurons that are tuned to a particular direction in 3-dimensional space, there or there, etc. Now came the surprise. We did an experiment also where the animal was inverted. They like to be inverted a lot of their time. So we had, in fact, 3 sessions, 2 upright sessions, in the middle an inverted session. And this is what we got. These are, again, 3 neurons, the azimuth tuning of 3 neurons. And what you see is that let's say, cell 1 is tuned in the same way to the same preferred direction in session 1 and in the upright session, in the repeated upright session. But in the inverted session, it's tuned to something else, to a different, it's still tuned, but to a different angle. And same for the other two cells. And if you look at it for a little while, you'll see that what happens here is that this is shifted by 180 degrees. Okay, so if you shift by 180 degrees, then these curves align. And this is not just these examples, it's across the population. On the left, you see the difference in preferred head directions for the two upright sessions. The difference is 0, so there's stability across sessions. And on the right is between the upright and the inverted session. And the difference is, from most of the cells, is plus minus 180 degrees, roughly. So we interpret it as evidence for a toroidal representation of 3D head direction. So on a toroidal representation, you have 360 degrees of azimuth, this is in blue, but also 360 degree of pitch in red. So in a toroidal coordinate frame, in fact, there is no inversion. It's the same azimuth all along. And why would something weird like that be happening? So the argument would be that if you have a standard spherical coordinate system for representing 3D head direction, then if I'm a head direction cell and I like to fire in that direction, the animal starts to pitch its head now. Okay, so I'm at 89 degrees pitch, I'm still firing because that's my preferred direction. And then I do a little bit further, I'm supposed to stop firing because I switch to this other direction. So there's a discontinuity in the zenith, and same below. So something that's non-desirable biologically, whereas for a toroidal representation, there's continuity, so that solves this problem. And indeed, the data are consistent. If you compare the firing in the upright and inverted conditions, the data are consistent with the toroidal coordinate system, not with the spherical one. And again, I don't have time to go into the details of additional experiments that Arseny has done. He had bats actually crawl on the inside, do a full 360-degree pitch maneuver in crawling. He's done also a flight experiment. It's all been published already, and all of these data are consistent with toroidal representation. Just to show you that this is not just a kind of mathematical argumentation, it has nothing to do with reality, show you how bats actually behave. You see that when they're landing, this is slowed down 10 times. When a bat lands on a ceiling, it goes through in this case, right now, it went through a 90-degree pitch maneuver. Okay, so it's an animal that flies in 3D especially, this is a real problem if it had a discontinuity at that point. And sometimes they're even doing full 360-degree maneuvers. So having a continuous representation would be useful. That would be the argument. Okay, so I think you got the point. We'll have one more landing, and then we'll continue. Okay, so there is a continuity. The toroidal representation is continuous. Also, it solves the non-commutativity issue, and the way it works is that the animals, these bats, tend to not roll their heads so much. And then if you have an animal that behaviorally, you have a dual trick here. It's generally 3D directions are non-commutative, meaning the order of your turns matters, and this is problematic if you want to make computations. Really, the order of how you get somewhere in its 3D directional spaces would determine where you end up. Whereas if the animal does not roll much behaviorally and the tuning is represented on a torus neurally, so which is two independent rings, then by definition it's commutative, which is another advantage of toroidal representation. It might explain why a lot of animals seem to not like to roll a lot. In fact, insects also apparently don't roll a lot. So I think this is an interesting interpretation of why a lot of animals do not roll. This would solve a non-commutativity problem of 3D directions. And until very recently, I would have this slide that like how much of this is relevant all to other species. Bats, of course, fly in 3D. And I was very happy. A few months ago, some of us were on this INAV conference. And Dora Angelaki, I was very excited to see Dora Angelaki and David Dickman give talks that show similar results from both monkeys and mice, that they found neurons tuned to 3D directions, particularly pitch and roll, including in the inverted positions in the mice, and stronger tuning to pitch than to roll. So they didn't do all that we did, but at least some of the things was very similar to what we find in the bat. So certainly monkeys that are very much navigating through, moving in 3D, and also mice that are perfectly fine with being with hanging upside down. I think it's interesting that they also have 3D compass in their brains. All right, let me move to the other story that I wanted to tell you about today, the goal-directed navigation using vectorial representation. So this is work by Ayelet Sarel, a very talented student in the lab, and also involved with the involvement of Arseny Finkelstein. So the basic question is, how do you navigate from point A to point B, right? So the issue is that all these cell types that we've heard about today, that we all know about, are mainly concerned with point A. Okay, we have place cells that tell me where I am, point A, grid cells that tell me maybe where I am, maybe distances. There are the head direction cells that tell me something about, let's call it north or nominal north. But where is point B, right? I know that there is the entrance to the building. I don't see it, but I know it's there. So somewhere in my brain, there should be a representation of where is the entrance to the building. It's point B. And I don't think we understand very well how this works. And just to show you that bats know where their goals are, this is our data that we recorded the GPS from an individual bat that we tracked for 75 nights. I'm just showing you a small subset of these nights. It goes from the cave to the tree on the left for many nights, then switches to this other tree on the right. And then on night number 33 in cyan, you see it's doing something very interesting. It starts in the direction of tree 1 and then changes its mind in the middle and goes to tree 2. And we have other nights when it did all sorts of shortcuts to yet other trees. So very interesting data, but it's just not quantitative analysis, just to make the point that they know where the goals are and that they can't be able to see those trees from that distance. So how does that work? Potentially, they could have a representation of literally a vector, a direction and distance, or certainly the direction will be the most important thing to their goal. So to test this, what I did is the following simple experiment. There is a goal in the middle of the room, just a platform with food where the animal has to fly around and around randomly and then land on the goal. And we record from CA1, and we define this goal direction angle, which is the angle between the direction to the goal and the heading direction of the bat. So in this case, when the bat is flying roughly towards the goal direction angle is small. And then the question is, are there neurons that are tuned to goal direction? And surprisingly enough, the answer is yes. So here are examples. So at the top are neurons that are tuned roughly to goal direction 0, so just when the animal is flying towards the goal. And at the bottom are neurons that are tuned, but to non-zero angles. And this is a distribution of preferred goal directions. You see that it's, first of all, it covers all possible angles, okay, which is good, but it's clearly an overrepresentation of goal direction 0. Okay, so flat plus a big overrepresentation at 0. So the goal direction tuning is not explained by place tuning. I don't have time enough to explain you all the analysis we did to show this, but just to illustrate. First of all, you see neurons with clear goal direction tuning, but with very weak place tuning, like this one. Of course, we also see the converse, so I can rest assured that we see plenty of place cells. In fact, they're about double the number of place cells than of these goal direction cells. So we have neurons like the one on the bottom that are tuned to position, but not to goal direction, but they're also the ones like the ones on the top. And there are a lot of cells with a conjunctive tuning to goal direction and to place, like this one. And then we can define the region of the around the place field and look at the goal direction tuning inside the place fields in orange and outside in gray. And you see they're rather similar in this neuron. In the second neuron, there's hardly any spikes outside the place field, but those few spikes that are fired there, it's the same goal direction as inside, and same for the third neuron. So you can compute the difference between the goal direction inside and outside of the place field, and they're around 0. So overall, we have 58 neurons from 3 bats, which were classified as goal direction cells. It's 19% of our neurons, so it's a sizable fraction. And they're defined by a high and significant directionality compared to shuffle data. And stable goal direction tuning, and they're we did a similar construction analysis to what we've just heard in the previous talk, where we assume that the neuron is a pure place cell, and how much can that explain the goal direction tuning, or vice versa. So we require for this to be defined a goal direction tuning. It's better explained by the goal direction tuning. And the goal direction tuning is stable across time. This is an example of 3 neurons along the flight of the animal. You see that if you kind of look at the different rows are tunings along the flight. The tuning is stable across the flight, not just at the very end that becomes tuned. So this shows the stability along the flight. Crucially, we asked, is this representation it's sensory-based because it sees the goal, or is it memory-based? And so to address this, what the editor has done is the following experiment. In the first session, so there was a curtain, an opaque curtain in the room, and the first session, the goal was behind the curtain. So the bat would fly in the central region, then from time to time it would fly behind and land. So when it was here, in the central region, it knew where the goal was. The goal was every day in a different location behind the curtain, but it did not see it. And then in the second session, we moved it to the central position. And so you see here examples of 3 neurons in the red, which are clearly tuned to the direction of the goal that's not seen, that's behind the curtain. And these neurons pretty much shut down or become much less tuned to the same location in space when the goal is not there. This is the bottom row of in red. And conversely, a neuron tuned to the central location is firing strongly when the goal is there, but not when the goal is not there. These are examples of 3 sessions. They're difficult to record 3 sessions flying bats and hold the cells and have the animal behave. But Yale was able to record some neurons. And again, you see this clearly preference to fire when the goal is in that location. This summarizes the data. This is an index comparing the activity to the tuning to the same location when the goal is there versus when it's not there and it's clearly skewed to the right. So the goal direction representation is specific to the goal. There are also interesting dynamics that I'll skip just to show that sometimes the cells switch very fast, but sometimes they switch slowly, like this neuron that when the goal was moved from the hidden position to the center position, for 10 or 15 minutes, this neuron continued to fire to where the goal was before. So it's a memory trace of where the goal was supposed to be. They're also tuning to distance. So you see here examples of 6 neurons on the left neurons. This is a raster of the various flights. You see it's this 2 neurons on the left are tuned to, are active when the animal is landing, distance 0, but these 2 neurons on the right are firing 7 meters before it landed. So at long path distances before landing. So this seems to be encoding the distance, the path distance towards the goal, and this shows the distribution of preferred distance of the distribution of the distance tuning curves in these neurons. So you see there's clearly a lot of neurons tuned to short distances, less than 3 meters, but there's also about 40% of the neurons, the ones below this line, are tuned to distances 3 meters and above away from the goal, like similar to the neurons on the right. And a lot of I think I'm running out of time, so I'll skip that. Just to say that many neurons are tuned conjunctively to the goal direction, goal distance. Like that cell is tuned to goal direction 0, but it's also when you look back along the distance, you see the firing rate goes down. So this is conjunctively representing the goal direction and the distance to the goal. So to summarize, we found in the hippocampal area C1 a subpopulation of neurons that are tuned to goal direction. The results from a curtain experiment suggest the representation is memory-based, not just sensory. We found interesting dynamics. There were goal distance cells, and the goal direction, goal distance signals suggest the existence of a vectorial representation that could support goal direction navigation. And you might speculate that this could be a neural base of the home vector, this notion that has been proposed for many behaviorally, for many animals, from insects to birds to rodents to humans, that a lot of animals can compute vectors to locations in space. Here are neurons that seem to do that. Okay, I'll skip what happens in large scale, just to say that we've constructed a very long tunnel that we're using now to address this 200-meter tunnel, moving on to do a 1-kilometer tunnel and all sorts of mazes that look like that, but I'll tell you about this some other time. So let me summarize. Why do we study bats? They have this sense of sonar that I didn't have time to talk about at all, but it's interesting to know that we can put a microphone and record with a millisecond precision the timing of the sensory inputs to the system, which is, I would say, it's difficult in a freely moving rat to record exactly when did it touch something or sniff something or see something. And here we have a millisecond precision. It turns out to affect very strongly the place cells. I didn't have time to show you that, but we've published this in the past. We can study 3D. Theta oscillation happens to be different in bats, so this allows us to constrain the spatial theories of hippocampal and neuronal function. They have excellent spatial memory. We'd like to bridge the gap between behavior in the lab and natural behaviors in the field. And the 2 most important points for me, one is comparative systems neuroscience. I think, at least as a sanity check, we should work in other species, not just in rodents, to see what does generalize and what doesn't. And I think it's interesting that some things are very similar, but some are very different. So I think it helps us to understand the invariant properties of the system. And secondly, the issue of natural behavior versus reductionism. I'm in a bit of the extreme coming from the neuroethological approach. I think it's really important to go with the natural behavior of the animals. So in our case, think about navigation towards goal, complex environments, 3D environments, large scale. So this is where we are heading with our research. So I'd like to thank the people in the lab, alumni, collaborators, funding sources, and thank you for listening.
Thank you, Nachum.